Platelets are best known for their role in hemostasis. They stop us all from bleeding, and they're also involved in the propensity for blood clotting. They do this by secreting various proteins when they get activated, and they also flip their membrane so that they serve as the surface for the assembly of the coagulation cascade and generation of thrombin, the most important clotting protein in the human body. So given these other functions of platelets, that they store cytokines and other proteins, they actually release them when they get activated. And this is relevant for cancer biology because platelets are known to stick to cancer cells, and then when they get activated, they release these cytoplasmic constituents locally. And those include factors that are known to enhance tumor metastases and spreading, such as various growth factors, platelet-derived growth factor, fibroblast growth factor, and VEGF, a potent stimulus for new blood formation. So the functions are beyond that of just platelet number. They also have distinct roles in enhancing tumor metastases, unrelated to their function, unrelated to their number, excuse me. So most platelet functional tests use activation-dependent assays. So release of p-selectin on the cell surface as a biomarker that can be very easily quantified by flow cytometry. Release of phosphatidylserine exposure, again by flow cytometry. But none of these are specific for tumors in cancer biology. So over the years, people have tried to move into, try to identify better biomarkers that can be linked to cancer. And the field has been focusing on genetics as a tool to further characterize biomarkers in platelets that may give links or hints to cancer. So when megakaryocytes generate platelets, the megakaryocytes invest into those platelets RNAs, microRNAs, and these can actually be quantified as novel genetic biomarkers linked to certain cancers. So these biomarkers these changes in the genetic architecture of a platelet have now been identified in a number of malignancies, including breast cancer, prostate cancer, colon cancer. And so it's fascinating to observe that platelets actually change their genetic composition when there's malignant cells nearby. And the mechanism for this is actually fascinating, and it suggests that there's a direct genetic transfer between cancer cells, so the RNA makeup of a cancer cell, into a platelet. And that appears to be how these platelets change their genetic composition. So again, it's very clear from a number of laboratories that if you take platelets from patients with various malignancies and you do genetic analysis of these microRNAs and these RNAs, you can actually see fundamental differences in a subset of these genes. These can then be quantified by sophisticated genetic assays to look at the qualitative changes and to actually quantify these biomarkers in platelets. So the advantage of the platelets is that they appear to store, they're enriched in these biomarkers. A lot of these biomarkers in cancers are measured in serum or plasma, and so we're missing a rich source of novel biomarkers that we now can identify in blood platelets. And they can be identified by routine genetic assays. So I think what's exciting about the field is the observation that that there is this change in genetic composition of these biomarkers. I think where the field is moving is, first of all, validation by other laboratories that these biomarker subsets exist. There has to be a standardization for assay development so that one can compare and quantify from sample to sample the expression patterns of these biomarkers. And a lot of these genetic quantitative techniques, especially when it comes to RNA abundance, are highly sophisticated. And so the next big step is to start to standardize these isolation methods and to quantify them so that clinical laboratories can start assaying in a meaningful format.